I'm Peter Adams, and I'm a professor of epigenetics and cell senescence and cancer and aging at Sanford, Burnham, Prebys Medical Discovery Institute in San Diego. And my role within SenNet is that I'm the MPI, so the multiple principal investigator, that's short for the MPI, together with Bing Ren at UCSD. The two of us are the leaders of the San Diego Tissue Mapping Center within San Diego. What we have been tasked to do within SenNet is to look at 5 different tissues in 2 different lines of mice. Specifically, we're looking at liver, mammary, hippocampus within the brain, colon, and bone marrow. We're looking at young mice, typically about 4 months old, and then old mice, typically about 22 months old. And what we're doing is to use very specific molecular markers of these senescent cells to really identify where do they accumulate within tissues and what do they look like. And our goal is to really kind of define that across the lifespan of the mice. Okay, so comparing the young mice to the old mice to see how these senescent cells accumulate in time and space across the lifespan of the mice. At the moment, we have most data from the hippocampus and the liver of the mice. We're starting with those 2 tissues. And what we have found is that we are seeing accumulation of these senescent cells in the older mice compared to the young mice. And we are starting to build up specific molecular signatures as to what those cells actually look like. Okay, there's been, over the years, there's been a lot of debate as to what a senescent cell, actually is. What does it really look like at the molecular level? And so we are now starting to get more of a handle on what those cells look like. So that allows us to better define them and identify them within the tissues. And what we're also finding is that, in kind of spatially, it seems that, these cells they're not randomly distributed within tissues. It appears to be that there are certain cell types which are more likely to become senescent than others. It's not that all cells become senescent as the mouse ages. There are some cells which appear to be more likely to become senescent. So it's worth appreciating that, within all of our tissues, okay, within humans and mice and most other organisms, the different tissues, whether that's the heart or the lung or the liver, okay, the liver in our case is a tissue that we're very interested in. The liver is made up of many different cell types, okay? The reason that it manages to do what it does as an organ is because it has many different cell types which make up that tissue. And so what we're finding is that, not all of those cell types are equally prone to becoming senescent. There's some more likely than others. We're also finding that there may be some non-random spatial distribution to these senescent cells. Perhaps they are clustering within certain regions of the tissue. Okay, they appear to perhaps concentrate together rather than just being kind of randomly distributed throughout the tissue. We need to really figure out ways to really age healthier. Okay, so that people don't, for example, have 20 years of chronic disease at the ends of their life. Maybe, they may only have a couple of years of chronic disease at the end of their life. And I think SenNet is contributing to, or will contribute to, allowing us to live healthier in that way. And so I think, we need junior investigators and, everything from junior principal investigators to postdocs and students and the cusp students. We need all of these people to be involved so that we can have the full breadth of ideas and and expertise and experience, and also so that we can train the next generation of scientists moving forward, so that we can continue to to tackle this challenge and overcome these problems. I'm really excited and delighted and very grateful to be part of SenNet. So often science feels like it's kind of competitive. We're all beavering our way in our own labs, trying to build up exciting stories and perhaps publish them before somebody else does. Okay, what I really like about SenNet is that it's so collaborative. Okay, when we get together for the in-person meetings, people are really genuinely sharing experiences and trying to help each other out. And these efforts that we have to to compare platforms together and even build up common panels of reagents that we can share with each other. It's just I really enjoy being part of that collaborative scientific experience. So I think it's also I mean, apart from what I've said about it being a tremendously important problem, I think it's really enjoyable to be working within SenNet. So yeah, I'm very grateful to be given this opportunity.